Right. This is G.C.E. A-level biology revision class. I have selected this question for today's class. Let's see the question. Which of the following statements regarding the digestion of nucleic acid in food in man is correct? So, what we're going to revise here, the digestion of nucleic acids. I will take this question again after revising the digestion of nucleic acids. Right. So let's starting with nucleic acids. Okay. I think you know very well what is nucleic acids. So nucleic acids, we have a two type, deoxyribonucleic acid, and the other one is ribonucleic acid, DNA and RNA. So if we eat something that could be a food with plant origin, a food with animal origin. Now let's say I'm eating a sugar, just normal sugar. This sugar is a extracted form from the plant. But if I eat a fruit or maybe vegetable, this fruit or vegetable is composed of plant cell. I'm eating a fish, I'm eating chicken or any meat. So this food consists of animal cells. Right. Then you know, either plant cell or animal cell, both have nucleus. So this nucleus, we can find large amount of DNA. And also, we can find RNA. That could be in the cytoplasm, example, messenger RNA, tRNA, or rRNA. Or maybe it could be in the nucleus, because nucleus produce RNA. Right. Now, when I eat this food, what's going to happen? During the process of digestion, these cells are open up and then contents released to the outside. So I'm drawing these things. So this is a DNA molecule and this is a RNA molecule. Now you understand, once we eat food consists of cells, the DNA and RNA released into the digestive system. Okay, now when the food passing to the small intestine, so this is the stomach, and this is the duodenum area of the small intestine. And the pancreatic duct connect the pancreas to the duodenum. Right, now pancreas secretes enzymes, many enzymes. But we are interested in a kind of enzyme called nucleases. So pancreas secretes these nucleases. So these nucleases are two types. One is nucleases act on the level of DNA molecule. So these are called deoxyribonucleases. Nucleases which are act on the RNA level, ribonucleases. Nucleases act on the DNA level, that means DNA is a substrate, enzyme is deoxyribonucleases. Then the substrate is RNA, enzyme is ribonucleases. So if these enzymes digest this molecule, you know already, DNA molecule consists of two strands, we call it double helix. But RNA strand consists of single strand. And you know very clearly, these DNA or RNA made up of nucleotide. Nucleotide. So how it looks like, it has this pentose sugar, either deoxyribose or ribose. And this has a nitrogenous base and the phosphate group. That's a nucleotide. So this nucleotide is the monomer or building block of this macromolecule DNA and RNA. And if we're going to look how this enzyme act now, I'm going to show in another view because I have deleted this one. I'm deleting this. So that's a nitrogenous base. And this one is the phosphate group. The phosphate makes a bond with the pentose sugar. So we know this position is C1, it's carbon 2, carbon 3, carbon 4, carbon 5, and 1 prime, 2 prime, 3 prime, 4 prime, 5 prime position of the pentose sugar. And this bond is called phosphomonoester. This is the phosphomonoester bond. When nucleotide join with another nucleotide, like this phosphate make another bond with the 3 prime position of the other pentose sugar. So then it's called phosphodiester bond. So this is the phosphodiester bond because there are 2 bonds. So we call it diester bond. This is a 1 bond. This is a 1 bond. Now these ribonucleases and deoxyribonucleases, they're going to break this bond. So when it breaks this bond, the 2 enzymes, deoxyribonucleases or ribonucleases, produce nucleotide. So I make a conclusion now. Pancreas secretes deoxyribonuclease and ribonuclease. Action of this enzyme, deoxyribonuclease, acts on DNA level and make nucleotide. Ribonucleases act on RNA level and make nucleotide. Clear? Right, now let's going to see what's going to happen to this nucleotide. at nitrogenous base and the phosphate. Now, second group of enzyme coming up, it's called nucleotidase. So nucleotidase act on nucleotide. So nucleotide is the substrate, nucleotidase is the enzyme. So this nucleotidase breaks upon the monophosphomonoester bond. That means these bonds breaking. Resulted phosphate group plus this one is now called nucleoside. Nucleoside. Then the third group of enzyme is coming, nucleosidase. So nucleosidase is enzyme, substrate is nucleoside. But it does break this bond. This result pentose sugar and the nitrogenous base. Finally, phosphate, pentose sugar, nitrogenous base. So these are small molecules, soluble small molecule, intestinal cell can absorb these molecules. So conclusion, nucleotides are further digested by nucleotidase to make the phosphate moiety or phosphate ion and the nucleoside. Then the third one, nucleosidase, digests nucleoside to make pentose sugar and the nitrogenous bases. Clear? Fine. There is another enzyme which is not much described. I also mentioned here that enzyme, maybe you need this knowledge of that enzyme. So this enzyme is called phosphatase. Sometimes it's also called intestinal alkaline phosphatase. Intestinal alkaline phosphatase. Now this enzyme, the major role is in case the cells also contain ATP-like substance, adenosine triphosphate, they are already formed, and these, they have 3 bonds of phosphate, 3 phosphate group. So phosphatase can break further these bonds to release the phosphate or phosphate moiety or phosphate ions from these nucleotides or ATP-like molecules. There is a recent research article published, and I have read this one. They said these kind of molecules can be pro-inflammatory, ATP-like molecules if we take from the food, and then phosphatase is good enzyme that can really break this phosphate and further leads breaking this apart, and then can absorb these molecules as its smallest molecular form. Right. Now we come back to the question again. Which of the following statement regarding the digestion of nucleic acid is correct? It starts in the stomach. This is wrong. I told you, start from the level of intestine, because pancreas releases enzyme, then they start in the process. Next one, DNA is broken down to nucleotides by nucleotidase. DNA is broken down to nucleotides by ribonucleases and deoxyribonucleases. They are called nucleases. So that's wrong. Nucleases break DNA into nucleotides. Third one, nucleoside is involved in the digestion of nitrogenous base. Now, what is function of nucleoside? Nucleoside breaks the bond between the sugar and the nitrogenous base. There is no digestion of nitrogenous base. So this is the nitrogenous base. This nitrogenous base is not digested by nucleoside. Nucleoside digests nucleosidase digests nucleoside. Fourth one, RNA is broken down to nucleotides by pancreatic nuclease. Correct. RNA broken down to its monomers, that is nucleotides, by nucleases, that is exactly ribonucleases. So this is correct. And the fifth one, intestinal nucleotidase act on nitrogenous base. So nucleotidase act on nucleotides to remove this phosphate, not on nitrogenous base. So we can take fourth one is the right answer for this question. Okay, if you enjoyed this lesson, give us a like, and I will upload more videos about next questions on my site.